Alright, this is our introduction to fungi. There are 4 fungi you need to remember for the midterm. The first one is Saccharomyces cerevisiae, Aspergillus niger, Penicillium notatum, and Rhizopius stolonifer. I'll show you guys how to identify each one of those as we go along. The first thing you have to know is the difference between fungi and bacteria. Bacteria are all prokaryotes. They have cell structure that's simple. They don't have a nucleus. Everything's just kind of in the cytoplasm hanging out, whereas fungi are eukaryotic. They have cell structure like us. They don't have any kind of, an enveloped nucleus. They have Golgi. They have all sorts of organelles that are involved in handling the production of proteins and things like that. Bacteria preferentially grow on PDA, whereas fungi grow preferentially on PDA, whereas bacteria grow preferentially on TSA. Key difference we'll talk about here in just a second. Bacteria have very simple cell membranes. They don't have any sterols, whereas fungi have more complex cell membranes involving sterols, having a more rigid structure. Bacteria also have peptidoglycans in their cell wall, which is kind of a random assortment of sugars attached to proteins, whereas fungi have very specific sugar arrangements, including glucans, mannans, and chitins. So they have a very specific type of structure to their cellular proteins. Mycology is the study of fungi. We know that all fungi are eukaryotic. They're all non-photosynthetic. They're also all chemoheterotrophs or saprophytes, so that means they burn chemical energy with oxygen like we do, or they can burn it with oxygen. They all secrete extracellular enzymes, which is unique. Not all bacteria do this, but all fungi do this. They have the ability to do that. Fungi fall into one of 2 categories, either yeast or mold. The difference is pretty easy. Yeast are all unicellular, so they have individual cells, look a lot like bacteria, whereas all mold are multicellular. They have discrete structures and functions for those structures. We also know that all mold are aerobes, so they require oxygen to survive, whereas yeast are facultative anaerobes. Yeast also reproduce by budding, kind of like bacteria. They kind of create a little bit of themselves and pinch it off and create a new little cell that way, whereas mold reproduce through asexual and sexual methods. One of the secondary structures of mold is they have these things called hyphae, which give them height, 3-dimensional structure, whereas yeast look a lot like colonies of bacteria on a plate. So when you see yeast growing on a plate, it tends to look a lot more like just regular old colonies. There are mycotic infections. You don't really need to memorize these, but you should be aware of them as they're growing more and more important in the global health issues. A really common one that most people know about is something like thrush, which is an example right here that babies usually get this, or people with compromised immune systems tend to have a problem with thrush. Athlete's foot is another one. Ringworm is another word for this right here. You can see an example right there on the skin. The more complicated ones that are less common are histoplasmosis and coccidioidomycosis. You can see an example of a histology stain for histoplasmosis right here. These become a lot more of an issue later on, though. Mycotic infections tend to target people who have compromised immune systems. So, for example, people that have something like immunodeficiency syndromes are very likely to get them, and they become much more severe. A great example of this is someplace like, say, where you have a lot of natural disasters, so Southeast Asia, which has a really high population of people with HIV, as well as a high likelihood for natural disasters like hurricanes, tsunamis, things like that. And you'll see an uptick of fungal infections, soft tissue fungal infections, and those actually have a very high lethality rate, nearly 100%. Basically, if you get fungi in your soft tissues, you have a very high likelihood of dying, and they're very difficult to treat because fungi have cell structures similar to humans, so they're very difficult to treat effectively. There's very few drugs that work on them. We grow our fungi on this stuff called potato dextrose agar. It's actually really simple. It's basically a bunch of dehydrated potatoes, a bunch of sugar on some agar. You could actually just make this at home. This is a really easy recipe in terms of media, but what it does is it creates a high starch, high sugar content environment, which creates high osmotic pressure. Bacteria do not cope well with high osmotic pressure, so they don't really want to grow on that. It's also slightly acidic, and bacteria by and large don't like acidic things, whereas fungi are perfectly fine growing in both of these conditions, so they favor fungal growth over bacterial growth. The reason why we do this is because bacteria are so proliferous in the environment, they even grow on the fungi themselves. What you'll end up having is an issue where if you try to grow fungi on a TSA plate, they won't grow effectively because the bacteria will overtake them and cause a lot of problems, so you'll have a lot of contamination. If you grow it on a PDA plate, though, you basically assure yourself that you're only going to get fungi growing on that plate. First one is Saccharomyces cerevisiae. It's the yeast found in grocery stores. You could buy it in a little packet. It's used to make things like bread, wine, beer, all sorts of fermented things. It ferments sugars into ethanol and carbon dioxide. This one's unique because it's one of the few fungi with a whole genome map. It was one of our first things that we mapped completely. It's used to do genetically engineered products. A specific one that you should be aware of is things like the hepatitis B vaccine. It's very useful. We used it to study a lot of different things in cellular structures because they have similar cellular structure like us. And they're also single cellular organisms, so they're a lot easier to manipulate than a whole animal or something to that effect. So here's an example on a plate. You can see it right here. It grows and looks a lot like bacteria on a plate, so that's how you know that you're looking at a yeast. And if you're looking at a yeast, you should say, oh yes, this is Saccharomyces cerevisiae, because it's the only yeast in the 4 organisms you have to know for this. Under a microscope, it looks like a lot like bacteria, but if you notice on 400x, these cells are much, much larger. You can see an example of budding right there. And over here as well, you can see where it's kind of pinching off a little copy of itself, and that's how it reproduces. But you can see that they're very, very tiny. They look like really tiny cells, but if you notice, this is 400x, so they're actually much, much larger than bacteria. The other one that we're going to look at is Aspergillus niger. It is a mold. It's used to make things like citric acid for food and sodas. It has these things called septate hyphae, which look like bamboo. They have little tick marks in them. And then there's a set of words that describe the way fungi look and that mold look, and this is a little bit technical, but we'll go over each one individually here. But what you should know is that conidiaphores are the things that sit on the surface of the agar holding the heads of the mold, which I'll show you in a second. And at the head of the mold, you'll see the black conidia or conidia spores that are produced in a chain at the end. So when you look at it, Aspergillus niger has this dark, gritty black coloration from it, the spores. So the conidia is what we refer to them as instead of spores, or conidia spores. And it's got this gritty black texture to the surface, so they're really easy to identify. Under the microscope, you can see they've got these really dark black conidia that are part of these multinucleate globous heads. You can see, it's kind of difficult to see the tick marks in these pictures, but you can see them somewhat here. These are the septate hyphae, and you can see the tick marks in them so that they are septate. The conidiaphore, though, is a special thing. The conidiaphore is the septate hyphae that holds up a multinucleate globous head. So this whole structure that holds up this head is called the conidiaphore. The trick here is that not all septate hyphae are conidiaphores. Some septate hyphae just transport nutrients. These ones specifically hold up these multinucleate globous heads. A good analogy for this is like a tree. A branch is made up of wood and it holds up leaves, but not all things that are made of wood are branches. It could be the tree trunk or the roots. So that would be an example to kind of help explain that. Multinucleate globous heads, you can see here, you can see the little black granules of the conidia. And if you go even closer, you can see underneath that, so here's an example here, you can see this stuff called the sterigmata, which produces the conidia. It's very hard to see. It's a little bit easier to see on an electron micrograph. You can see right here, these sterigmata that produce these globus conidia in chains at the end. So this is what it looks like in actuality. You can see that they've got little lines right there. So that's what it actually looks like. So sterigmata grow at the end of the conidiaphore and produce the conidia from the sterigmata. The next one we're going to look at is Penicillium notatum. It's the mold that produces penicillin. It is a low white septate hyphae. It kind of looks like kind of a white fuzz. It tends to grow on citrus fruits and soil. So if you ever see an orange with kind of like that blue-green and maybe a little white topping to it of mold, that would be most likely Penicillium. The conidiaphore in this one branches like a fan. I'll show you what that looks like in just a second. And it produces these blue-green conidia on it. The Penicillium cultures are injected into blue cheese to produce the blue veins. Not Penicillium notatum that does this, but other Penicillium cultures are used to do this kind of production. So it tends to have a blue-green color. Now this isn't a great picture, but what it does show us is an example of the extracellular enzymes. The enzymes here actually change the color of the agar to this nice yellow coloration. And it does look kind of like this blue-green, but the images that you guys will see on your plates are actually a little bit better. But you can see that this is an example of Penicillium notatum because it's got the alteration to the agar and it's got the kind of got this blue-greenish coloration to it. Under a microscope, you can see it's very, very large. And you can see that it's got these nice septate hyphae. You can see the tick marks quite clearly here. So that means it's septate hyphae. This is the conidia-4 that holds up the head. You can see the sterigmata right here, which produce the globus conidia in chains. And it kind of looks a little like fingers. And I had a student once tell me it looks kind of like its name, ILLI, where it's got the long fingers with the little dots for the eyes on the end. And that's how they remembered that one to memorize it. So you can see the globus conidia in the chains, the sterigmata, and the conidia-4 with the septate hyphae with little tick marks on it. So that's pretty easy to identify. It looks a little bit different under an electron micrograph, but it's more or less the same. I like the pictures of the slides better. It's easier to identify. Okay, the last one is Rhizopus stolonifer. It's the common bread mold. It doesn't really produce anything of use. It's just a spoilage organism. One of the things it does do, though, is it produces really long white hyphae that will actually knock off the lid of a petri dish unless you tape it down. So it grows up really tall. It has hyphae, but these are non-septate, so it's like a straw. It's perfectly smooth. It doesn't have any tick marks. And of course, because scientists hate people learning things, they changed up the naming scheme for this. They've changed it to sporangia or sporangia spores. And I'll kind of go over these in just a second here, the definitions. But what you should know is that this one's unique because the spores are only released when the sporangium is damaged. So this one only releases its spores when it hits a scenario where it damages that spore packet at the top. Otherwise, it doesn't release them. So it's kind of its way of projecting itself only when there's damage done to it so it can move. This one is interesting because it has two structures in it that are in its name, rhizoids and stolons. Rhizoids are the root-like structure that attach it to the auger. It's not really sucking up nutrients, it's more anchoring to things, and I'll show you what that looks like in a second. Stolons, on the other hand, are these hyphae that run parallel to the auger, like Bermuda grass runner. A better example, I think, is also that if you think about a sprinkler head system, the sprinkler heads are like the heads have the spores on them, and underneath it are these PVC pipes that run parallel to the ground, transporting water and nutrients to the sprinkler heads. And that's what the function of the stolons are. They are there to transport nutrients across to one another. So Rhizopus stolonifer has two structures on it, rhizoids and stolons. So that's how you can identify it pretty easily. You can see right here, it's very thick brown growth. It actually knocked the lid off of this dish, so you have to be very careful with it. It produces spores that are brown in color. You can see that brown grittiness right here on the spore heads. These are called sporangia. They are held up by sporangia-4s, and the spore parts are called spores or sporangia spores, and they are in that thick, dense coat. Remember that the fun part about these ones is that this coat only releases spores when it gets damaged, and that way it can project the spores out into the environment. So it's very tightly packed, as you can see in this electron micrograph. And even under the slide, you can see it's very densely packed and it's got that brown texture to it. This one has non-septate hyphae, so you can see this hyphae is very smooth. It's straw-like, it doesn't have those tick marks. And if you look right here, you can actually see an example of the rhizoid, which is this root-like structure. This is what it actually looks like in nature, so you can see it's kind of like a suction cup. It basically anchors it to whatever it's growing on. So that's what it looks like in reality. And it's pretty easy to differentiate it like that. If you see those root-like structures, you can say, oh yeah, that must be Rhizopus. All right, that covers our fungi. Thank you very much.